Thank you very much. Our next speaker is Ravi Jagasia. So thank you very much, Ravi, for being here. Ravi is lab head of human neurostem cell at Roche and project leader for small molecule drug discovery program for neurodegenerative therapy for Alzheimer and disease-modifying therapy for Parkinson. Prior to joining Roche, in his current role, he did a postdoc at the Helmholtz-Zentrum in Munich, focused on a model of central nervous system neurodegeneration and neurodegeneration of the nervous system in vitro model. During his PhD at Max Planck Institute for Neurobiology, Ravi demonstrated the importance of mitochondrial protein in developmental programmed cell death in vivo. Ravi holds a PhD from Adolf Bunternat Institute for Physiological Chemists at Ludwig-Maximilian University in Munich, and a Master and Bachelor of Science from the University of Toronto, Department of Physiology. So Ravi is here, and he will present from the IMI project EUAIMS, a European Autism Intervention, a multi-center study for developing new medications. So thank you very much, Ravi, and sorry for the mislead.
Thank you for the nice long introduction.
I tried to make it short.
No problem. Now I would like to expose you a bit to autism, to one facet of the EUAIMS collaboration. And EUAIMS goal is to drive basic preclinical clinical research in autism in Europe. And one aspect of this project is to use human pluripotent stem cells to model the disease. And I would like to explain to you today why this is important and the relevance of these cells in the context of basic understanding of autism and also drug discovery. So autism spectrum disorders is a group of neuronal developmental neuropsychiatric disorders characterized by early childhood deficits in three classic domains. So deficits in social communication, abilities of the acquisition of language and maintenance of language, and abnormal but severely debilitating repetitive restrictive behaviors. Neuropsychological deficits, also comorbid with these kind of core symptom domains of autism, include epilepsies, seizuring, attention deficit disorder, mood disorders, and as well also non-neurological complications such as gastrointestinal or immune dysfunction. Just a brief number, about 1 in 88 children are affected with ASD. And this actually represents 80% increase in the prevalence of ASD in the last 8 years alone. And this is hotly discussed and debated, the reasons for this in the autism community. Currently, there are no effective treatments to basically treat these core domains of autism. So research into autism is highly relevant. However, this is a very good opportunity, a very good time in autism research. So with the expansion of human genome projects, the ease of DNA sequencing technologies, there's starting to become a very good understanding of at least some of the genetic causes of the diseases. So ASDs are caused by both environmental and genetic, but there's a very strong genetic contribution here. So twin studies, sibling studies can estimate up to 70% of the disease can be accounted for by heritability. Early on, there was the identification of single gene or monogenetic disorders that were highly penetrant causes of ASD, but with the advent of new sequencing and new studies, it's becoming clear that there's over 100, 200 plus genes implicated in the disorder. with, and there's also a bit of complexity, sorry about that, in the sense that sometimes it's clear that you might need multiple genetic hits to have an underlying pathophysiology of the disease. So, and a very important point, and also now a very big challenge in autism basic research is the fact that none of these genetic perturbations count for more than 1% of the total ASDs. So a big question in the field, which is still unresolved, is whether we're dealing with multiple diseases such as cancers or epilepsies, or whether these genetic perturbations can somehow converge on some common molecular, cellular, brain circuitry dysfunction phenotype of the disease. And one very important technology, as the previous speakers discussed, was human pluripotent stem cells, which can be now used to address this question. Why is that? And just to kind of glimpse here at different ways, basically experimental models to study ASD. On one hand, you can look at kind of human studies, the potential to do clinical trials, which are highly, highly human relevant. And then you have the possibility to study autism using mouse models by, for example, taking some of these single genes and knocking them out or disrupting them in the mouse if they're conserved, and looking for potential symptoms of autism. But given the fact that autism has a very complex genetic heterogeneity, actually now iPS cells, or human-induced pluripotent stem cells, give a very nice opportunity to basically model this complex genetic heterogeneity and try to understand on a molecular, cellular, and circuitry level what's going on in the disease. So there's a huge need in autism research for relevant human models of the disease. And the goal of this aspect of the EU AIMS project is to start to address, is there some sort of convergence in the context of autism disease biology using human pluripotent stem cells? So I mentioned a bit about this convergence on a potential kind of functional convergence, that even though you have very different genetic perturbations in the disease. And what is becoming clear is that there's one actually substructure or function of neurons in the brain that seem to be important. So several of these, for example, several of these monogenetic genes encoding for proteins in autism seem to play very important functions in synaptic physiology. So synapses are the point of communication from one neuron to the next, and kind of function of synapses are very important in the context of learning and memory, and clearly in circuitry relevant for autism, such as social memory. So there seems to be really some sort of synaptic dysfunction that could potentially underlie this disease. So the main goal of our the research efforts in IPS in EU AIMS is to basically take patient cells and ask, first of all, can we see this synaptic dysfunction, and is it, do we see common perturbations across multiple genetic groups? And the workflow that has been achieved for this within this project is actually recruiting ASD patients with both known genetic perturbations of the disease, as well as idiopathic patients, unknown causes of the patients. From the patients, taking actually, in this case, what is pretty novel is taking hair samples from the patients and then growing these up, banking them, from them reprogramming them into induced pluripotent stem cells. From there, then differentiating them down the neuronal lineage, effectively making brain tissue in a dish, and asking ourselves, can we see, for example, synaptic function in these neurons, and can we see actually alterations in the context of disease? And then with a bit of molecular and cellular understanding of potentially the disease pathophysiology through these cells, then use these kind of potential phenotypes in the context of drug screening and drug discovery, and to, at one hope, identify novel therapies for patients. So just currently, where are we with this project? What has been achieved? The project has been ongoing for 2 years. So multiple IPS lines have been generated from multiple patients, and also multiple genetically defined patient populations. Very robust protocols have been put in place across the laboratories involved in this project to generate neuronal precursor cells, then to subsequently differentiate them to human neurons, mature them to a level where you can study, for example, synaptic physiology and plasticity, such as LTP, LTD, long-term potentiation, long-term depression, which are correlates of memory and associated functions thought to be super relevant for autism. This has been achieved with at least 2 different genetically defined populations of ASD. It has been possible to identify molecular, cellular, and neurophysiological deficits in the cells. So with this in mind, and with this encouraging results, this is turning into a very promising avenue of research to understand autism, also the new expansions in the genes involved in autism, and currently in several of the FBA labs, including us at Roche, for example, we're starting our first high-throughput screen on a disease-relevant phenotype coming from patient cells. So that's what's been achieved. And with that in mind, I hope I gave you a bit of enthusiasm that there's huge opportunities of human pluripotent stem cells to understand ASD, autism disease biology, both from a basic understanding of disease mechanisms, the potential to identify new kind of drug targets in the disease, and also to be used in drug discovery. Just would briefly like to say that within this consortium, there are several open questions that we would like to address over the next 2 years. One is what I mentioned before. Is there a convergence of these multiple different genes involved in ASD onto a very specific phenotype? And so we're addressing this question by taking multiple genetically defined forms of the disease and asking, do we see similar phenotypes? Then another point is, with these phenotypes and this higher resolution understanding of disease progression, can we actually use this to drive drug discovery, identify new targets or potential biomarkers relevant in the context of the disease? And an important facet, and what makes this EU AIMS project rather unique, is part of the project is to run a clinical observational trial of ASD individuals. And from that, sampling of hair cells, of tissue, is ongoing to potentially extend our results beyond just a genetically defined population into an idiopathic unknown population. And the question is, can we actually identify relevant disease signatures in this population? So with that in mind, I would like to just briefly end and acknowledge the people involved in the EU AIMS work package involved in the human pluripotent stem cells. And this is driven by, actually, from the academic side, from Jack Price, professor at King's College London, and there's several FPA partners involved. And at Roche, I would like to just briefly acknowledge Will Sporin, who's the FPA coordinator for the EU AIMS project. Thank you.